Hello, fellow biologists, now fellow botanists at this point. Welcome to our second lecture on alga and the transition from algas to plants. So we'll start again with a UR here, and in our previous lecture we had talked a lot about archaeoplastids. So this is the group of algas that had first evolved chloroplasts. So these are the ancient chloroplasts. So on our tree, origin of plants over here, tree number 2, archaeoplastids are down here, and our archaeoplastida are the group of red and green algae that have the first chloroplasts. So we've talked about those. And we've also talked about several multicellular groups of algas. We talked about the phaeophytes, the brown algae, which are over here. We talked about the red algae, the rhodophyta, right here. And we also talked about the green algas. And remember that the green algas are actually not a monophyletic group. They're actually sort of a group that, while they have a common ancestor, this ancestor doesn't include a lot of other organisms, and these other organisms are going to be our vascular plants, as we're going to talk about in a minute. So we talked generally about the chlorophytes. So here are a bunch of chlorophytes over here, and one particular group of chlorophytes were the charophyceans, right here. And it seems likely that the charophyceans, at least a few of them, gave rise to our modern group of plants. And we're going to refer to our modern group of plants as embryophytes. And a little bit later in the lecture, we'll explain why we call our plants embryophytes. But these are our modern group of plants, our embryophytes right here. And they include the non-vascular plants, the bryophytes, and the vascular plants. And we'll get to those in the next lecture. But in this lecture, what we're going to do is we're going to talk about the transition to land. And so we're going to talk about how we make this transition from being an aquatic alga, an aquatic green alga, to actually being a plant that lives on land. And if there's sort of one thing that unites plant evolution, it's that plants have been evolving now for well over 400 million years in an effort to colonize land. Remember that all these algas come from water, and so the evolutionary impetus that drives so much of what we're going to see in plants is this evolution towards land. So in this lecture, we're going to talk about the problems that an ancestor of plants would face making this transition from an aquatic environment to a terrestrial environment, and then we're going to talk about some of the key adaptations. So I'll come up here. Whoops. I'll stay down here. Plants first colonized land about 475 million years ago. And so this may not seem like a big deal, but think about what land was like, what the terrestrial Earth environment was like before plants came. There were, of course, no animals on land. There were really probably not a lot of fungi on land. Maybe some sort of encrusting organisms, some encrusting bacteria. Maybe there were some green algas that sort of could deal with a drier environment, but that's about it. If you had come onto land, 500 million years ago, some sort of terrestrial environment, it really would have looked very, very barren until plants arrived. So, if you'll permit me a moment of silliness, plants were the first organisms to boldly go where no other multicellular photosynthesizing organism has gone before. Yes, right, you don't have to actually go into space to boldly go where no one has gone before. You could have been a plant 475 million years ago. All right. So, not only were plants then the first organisms that appear, first large multicellular organisms that appear on land, they also changed the trajectory of life on Earth in some very fundamental ways. And so, oops, I gotta keep moving over here. One of the quotes that I particularly like, these of course are for plant biologists, but they don't really mince their words over here. The colonization of land by plants is one of the most significant evolutionary events in the history of the planet. All right, so what makes the colonization of land so significant? And there are all sorts of reasons, but I'll highlight a couple. The first is that oxygen concentration significantly increased. If you now are colonizing land with plants, we have a lot more photosynthesizing organisms. These photosynthesizing organisms are reducing carbon dioxide in the atmosphere. They're putting in more oxygen in the atmosphere. And so our atmosphere is going to change very significantly as the result of plants colonizing land. And of course, if you have more oxygen in the atmosphere, that means that you can have very different animals that require a lot more oxygen for their metabolism as well. Okay. And of course, if you have plants on land now, you make land inhabitable for animals because of course animals need something to eat. They're the plants. And if you have plants dying and animals dying on land, of course now you can also have a lot of fungi and other decomposers that are going to break down all these dead organisms as well, which of course then is going to change the soil change the entire makeup of land. Instead of being fairly barren, we're going to have lots of soil. It's going to have lots of carbon in it, and so in fact the terrestrial world as we know it simply would not be possible if plants had not colonized land. So if you're glad and you enjoy being where you are today, thank your parents who've worked very hard to raise you, thank your teachers and coaches and mentors who have helped you along the way, and thank plants that colonized land. All right, so let's start considering some problems. What would happen if you took water away from algae? So to sort of put you in the right frame of mind over here, imagine that we've got some sort of algas over here. And admittedly, these are kelps. These are brown algas, but they'll serve. They'll give you an idea of what we're dealing with. And so they're doing very fine over here, swaying in the tides of this kelp, particular marine kelp forest. However, let's suppose you take away the water. And so here's a picture of a bunch of brown algas at a very low tide. And so it doesn't look sort of as beautiful as our first picture up here. All right, so what's going on? What are some problems that these organisms face? And first of all, they don't have a lot of structural support. In other words, they don't have support to be able to stand up in air. They can do just fine in water where they can float, but not so much in air. All right, so everything kind of looks like overcooked spinach. Secondly, transport is going to become a problem now. So in our little picture over here, notice that at least parts of our algas at low tide are still actually in water. All right, but there's now parts that are out of water. So there are parts of plants that might have access to nutrients and to the water, of course. There are other parts of plants that don't. And so if your organism now is existing between these two very different environments, you might have to move things between these environments, right? You might have to move nutrients up. Right? But a lot of your gas exchange now is more possible maybe in the air, right? So any sort of photosynthesis you're doing might be more possible in the air. Now you have to move carbohydrates down. All right, so you need to be able to do transport now as you start to have an organism that exists in very different environments. But our algas don't really have transport systems. They don't have transport tissues. All right. A third problem is with drying out, desiccation. Clearly we're in an environment now that is not a water environment, at least for part of the organism's bodies, and so they can dry out. Evaporation is happening. And finally, even if we could solve all these other problems up here, even if we could get sort of an upright organism that can transport whatever it needs to transport and that can avoid drying out, we're now out of water and we still can't reproduce. So if you manage to do all this, you still can't pass your genes on to the next generation. And of course, that is the end of the line, evolutionarily. It's very difficult to reproduce if you don't have water for your little sperm cells to be able to swim to your egg cells. All right, so we have a bunch of problems and we need to solve these problems. So let's look at some of the solutions that plants have come up with. And let's start with the problem of reproduction. Because as we mentioned, if you can't reproduce, then any other solution you come up with doesn't get passed to the next generation. And so what's going to be interesting about the plant life cycle is it's an adaptation to the problem of reproducing in a terrestrial environment, but it doesn't directly solve that problem, at least not initially. So what plants are going to do is they're going to alter the life cycle. And so the life cycle isn't going to mean that we suddenly don't need water at all, but what it's going to mean is that when you do have reproduction, we're going to make that reproduction count for a whole lot more. In other words, if fusing a sperm cell and an egg cell, fertilization is difficult, all right? When it happens, make it count. When you have sex, make it count. That's going to be the key to the plant life cycle. Right? So let's see how the plant life cycle is going to work. Remember, in our Chara life cycle, we had a multicellular gametophyte, a multicellular 1N generation, but we did not have a multicellular 2N generation. In the plants, what we're going to do, the big innovation of sorts, is that we're going to have a multicellular haploid and a multicellular diploid generation. In other words, we're going to alternate between multicellular generations, and so we're going to sort of refer to this life cycle in shorthand as the alternation of generations. And so every life cycle, of course, if you're going to have sexual reproduction with eukaryotic cells, alternates between haploid and diploid at some point, but we're alternating here between multicellular generations. All right, so where do we start? Let's start, as we did with Chara, let's start with the gametophyte. And remember, a gametophyte is a plant, a phyte, that produces gametes. It's multicellular. So here's our 1N gametophyte plant. And it, of course, is going to produce gametes. And remember, these gametes are going to be produced by mitosis. All right, so remember, gametes now are produced by mitosis, not by meiosis, as we've considered in the animal life cycle. So we have mitosis producing sperm cells and egg cells, and if a sperm cell fertilizes an egg cell, we get a zygote over here. And so far so good. That's what we had in the Chara life cycle. However, what we're going to do differently is that our zygote now is going to divide a whole bunch of times by mitosis to give us this new multicellular generation. All right, so what is this new multicellular generation going to do for us? Well, this new multicellular generation, after it gets bigger, is going to go through meiosis, and not just meiosis once, not just the zygote meiosis once, but lots and lots of cells can now go through meiosis and produce these structures over here that we refer to as spores. All right. So what kind of multicellular plant is this? Well, this is a phyte that produces spores. It's a sporophyte now to complement our gametophyte generation. So what's new, right? Our zygote now has produced a sporophyte, a 2N generation, and right, this sporophyte then undergoes meiosis to give us spores, which are 1N again, which produce our gametophyte. If this were all that the plant life cycle was doing differently, it would still be neat, but remember that there are a lot of algal life cycles that had an alternation of generations too. What makes this life cycle particular to the plants is that it's an alternation of generations, but with this very important innovation that Chara gave us. So what I once again have is our plant life cycle over here, but instead of just indicating the gametes, the spores, and the zygote, I've put them in their particular places where they'd be found on the gametophyte. And remember that Chara holds on to its egg. It has a gametangium, which is a structure that contains a gamete. This particular gametangium is an archegonium. It hangs onto an egg. And then, of course, there's another gametangium over here. It is an antheridium, and it contains sperm cells. So when we have a sperm cell that swims over here to fertilize the egg, right? Here's our fertilization event now. This cell has become a zygote. And as we've seen now, our zygote is going to divide by mitosis. So let's have this cell divide by mitosis over here, and we're going to start to produce a little plant that comes out of it over here. We'll even give it a little bit of a capsule over here. This plant is now our sporophyte. So notice what our gametophyte is doing. Our gametophyte is actually nurturing and taking care of our embryo sporophyte as it starts to grow, right? Little embryo sporophyte. And so, because this is something that all plants do, we refer to plants as embryophytes, right? Because the embryo sporophyte is nurtured by the gametophyte. And this is something that every single plant is going to do. whether it's a little moss, whether it's a giant redwood tree, whatever it is, the embryo sporophyte is going to be nurtured by the gametophyte. All right, and so that's why plants are called embryophytes. And of course, remember that this alternation of generations made possible by the nurturing of a sporophyte from the gametophyte is going to make meiosis more possible. We can do lots more meiosis with a multicellular sporophyte, and that is going to allow us to have many more spores. So when we do have this rare fertilization event over here, we can now get lots and lots of additional meiosis from it, and we can make lots and lots of additional spores from it. So I'll move up here to make this final point, and that is that all plants have this life cycle. And so as we go through and look at different life cycles of different groups of plants, you're perhaps going to be tempted to think, well, but there's a lot of variation, okay? We're tweaking the life cycle. We're in the sense that we're giving it new structures here and there, but every single plant life cycle is going to have this basic structure, this alternation of generation. So as a reminder, what are we doing? Let's compare what we are doing in our plant life cycle to what we did in the chara life cycle. So for chara, how many spores were produced per zygote? And the answer is we do meiosis once, so we get 4 spores. How many spores are produced per zygote per plant? And potentially you can just keep doing meiosis forever, literally forever, if a plant can live long enough. So the number here is now infinite. We go from 4 to infinite. So what is the advantage of a sporophyte generation? It vastly produces the number of spores that we can make from our fertilization event. All right, let's tackle our second problem now. And our second problem is actually 2 problems. So we're going to put them together, the problem of transport and the problem of support. But what I hope to show you is that if we solve transport, we also solve the support problem. All right, so what's the first solution? Well, one of the things that plants do that algas don't do is that plants have an apical meristem, and we'll talk more about apical meristems in a little bit. But the general idea behind an apical meristem is this group of undifferentiated cells that can sort of keep dividing and keep producing more new tissue. All right, and so plants have apical meristems in different places, but one particular meristem over here is the shoot apical meristem. So it's this region right here in a little cross section of a plant. So think of this as being like the bud of a plant over here. Right? And so this region of cells can divide, and it can produce a lot of new tissue. So we won't mention all of this over here, but what I want to mention is that it's producing, among other things, some tissue that is going to be transport tissue. All right, so specifically it's producing vascular tissue. So we can see little traces of vascular tissue here, little traces of vascular tissue over here. We can also see little traces of vascular tissue that are produced in developing leaves, and so on, right? So these are all little traces of transport tissue that are going to occur in plants. And this transport tissue is going to be produced by our apical meristem. So many plants have vascular tissue, not all, but I wanted to introduce you to the concept of vascular tissue and transport at this point, because we're going to look at a lot of variations on transport in the lectures ahead. So one particular type of vascular tissue is xylem. And the general idea behind xylem is that it's going to move water and it's going to move minerals. And so minerals would be things like nitrogen or phosphate or so on from this environment. And usually our water and our minerals are going to be found in the ground. So in general, right, very generally speaking, xylem is moving things up. Right, it's collecting all this from the ground, it's moving it up the stem and into the leaves and the body of the plant. All right, so we've got a transport that moves things up. Now, if we have a transport that moves things up, it turns out that we're going to need very thick walls in our cells and very sturdy walls. And so our very thick cell walls are going to be enforced by a compound that we refer to as lignin. All right, so if we look at lignin, over here, here is a plant cell, and you can see that it has a wall a primary wall and then a secondary wall. And so if we zoom in, here's the primary wall, here's the secondary wall. And in plants that have in xylem cells, I should say, these secondary walls are filled with a lot of lignin to make them really, really sturdy. And these thick walls are necessary, of course, for transport. And because they're necessary for transport, they also end up providing support. So let me explain why we need thick walls for transport and then how that gets us to support. You have a little cup of water here, very artfully drawn. Here's a little bit of water. And you want to move the water up through a straw. All right, and that's basically what plants are doing in xylem tissue. They're trying to move water up through a series of tiny little straws. So what you have to do, of course, is you have to provide a vacuum. All right, you have to lower the pressure inside the straw. So I'm going to put a bunch of little minuses over here to indicate that there's lower pressure inside the straw. All right, that's what you do when you suck up on a straw. Now, if there's lower pressure inside the straw than there is outside, and I'll put some pluses out here to indicate that we have higher pressure outside, what that means is that our water is actually pushed down and up into the straw. All right. So if you have a vacuum in the straw, water is actually pushed into the straw and moved up. All right. Now, of course, it doesn't take a whole lot of suction to move water up a little straw like you'd have in a little drink or something like that. But now think about plants, and plants are going to get taller and taller and taller. You're not just moving water up, a few inches. You're potentially moving water up feet, right? So let's extend our straws up here. Sorry about that. Straw's getting a little crinkly. And so the higher you want to move the water is moving up over here. The higher you're moving the water up, the more negative pressure you're going to have to apply, right? Lots and lots and lots of negative pressure to move the water up and to move it against gravity. Now, if you're really creating a negative pressure area inside your straw, if you just use your regular little plastic straw from, I don't know, a restaurant, McDonald's or something, that straw is going to collapse. So what do you do to keep that straw from collapsing? Well, what you need to do is you need to make your straw a lot thicker, right? You need to enforce the walls. So we're going to enforce the walls of our straw over here. And if you are using an actual, if you're using a human straw, you might enforce it with plastic. But if you're using a plant straw, what you're going to do is you're going to enforce it with lignin, right? You're going to take these cells that are moving water up. You're going to put a lot of lignin over here in these secondary cell walls. All right. So this lignin over here, this is what allows us to move water up and create a really high negative pressure area inside our xylem cells. Now, if we are going to strengthen our xylem straw, so to speak, to make this negative pressure possible, what that means is that our xylem cells are also able to provide a lot of support for our plant. So in other words, the idea is that transport leads to our solution for support as well, particularly in xylem. Right? And so ultimately what you see is for very, very big plants, such as a tree over here, if you take a cross section, most of what you're seeing in the cross section of the trunk of this tree is xylem. You see a little bit of other tissues to the outside, but like 90+% of what you're seeing over here is xylem, the secondary xylem over here. And so I brought along a little tree over here, and so if you look at this cross section of a tree, this is a willow as you can see, I'll turn it around so you don't have willows, notice that most of what we get in the cross section, all these things that are rings that you sort of count as tree rings to figure out how old the tree is, these are all xylem rings. And of course they're all very tough, which is why we can make a variety of things out of xylem, such as wood, furniture, and so on, right? These are very hard cells that are reinforced with lignin. All right, so xylem is one type of transport tissue, but let's not forget the other type of transport tissue, which is phloem. And so phloem is going to move photosynthates from the leaves, usually, where most photosynthesis happens. And because leaves generally tend to be higher on a plant, the idea is that phloem generally moves things down. So again, there are, of course, very obvious exceptions to this. For example, in the spring, when we have lots of phloem that has been stored in the roots, and the phloem is moving So turn over here, as you can see, and so this little line of phloem, when the phloem is moving sap back up from the roots into the stems to make new leaves in the spring, right? We can put a little tap inside our, just inside our outer bark, through the phloem, and so then as the phloem is moving as tissue up, all right, that sap drips out, and if we do this for maple trees and collect it and boil it down, we get maple sugar. So there certainly are exceptions to the idea that phloem only goes down, but as a rule of thumb, that's a good way to think about it. All right, so we've now solved three of our major problems. We've solved the reproductive problem, sort of. We solved transport and we solved support. But now let's think about our next one, which is drying out, desiccation. And let's first think about the desiccation of tissues, all right, the actual vegetative tissues. And so one of the best ways to keep water from leaving a structure, particularly a structure like a leaf, is to put some sort of wax around it, because wax is impermeable to water. And so that's what plants often are going to do. They're going to put some sort of waxy layer or cuticle around a leaf or a plant body in some form or another. All right, so I was looking for a good cuticle picture. There are not a lot of good cuticle pictures, so I had to make my own. And so there are, you can see a lot of leaves with really thick cuticles if you just take a step outside of Asher. So I brought a couple of different types of leaves. So one type of leaf, of course, is holly, right? Literally just outside the door of Asher. And if you take a few steps further, you can find lots of magnolia trees all over campus. And notice that these leaves are sort of really tough and leathery, both of them. And the reason that they're tough and leathery, among other, well, several reasons, but one of them, of course, is that they have this very, very thick cuticle. And holly leaves, and at least some species of magnolia, have leaves that last all year. And so this cuticle is very important for helping the leaves hold on to water, particularly during winter, actually, when it can be very dry and very difficult for plants to hold on to the water. And so what you can do, and what I did in this picture, is you can use a little razor blade or tweezers or something to scrape away some of the cuticle. And so notice there we sort of create this little you can see once I peel it away that this outer layer is clear. It's sort of this waxy layer. All right, so that's one way to see it. If you want to just do this on your own and you're not feeling that patient, what you can do is you can just sort of take a leaf like our holly leaf over here and rip it apart. Don't be gentle about it. Don't rip along the veins. Rip counter to the veins, and create some really jagged breaks over here. So if you can create some jagged breaks, I've got a pretty good jagged break right here, and then if you look at it crossways, you can see different layers. And the camera, of course, on this computer is not high quality enough that you can really see it, but you can see some cuticle layers at the top and a little bit at the bottom too, and this is the cuticle that we're talking about. All right, so wrap a leaf or wrap the plant body in some sort of structure that keeps water from being lost. However, right, for every solution, there's another problem. And the problem that we encounter is that if you wrap your plant body in wax and its cuticle so that water can't escape, you also don't allow other things like gases to get in. And of course, plants need gases like CO2 in order to photosynthesize. So we need another solution to our solution. And there are a couple of ways that plants allow gases to come in. So one is you just sort of punch a hole through the cuticle, and this is called a pore. It's a surface opening. And so in this stylized picture over here, you can see this opening through the waxy cuticle and the epidermis over here into the interior of the leaf. There's all these air chambers around our cells that are photosynthesizing, and so gas can just move freely through these pores. And here you see an actual pore in cross section. Here's the opening, right, that allows gas to move back and forth. All right, so having some openings is good, but that still doesn't allow us a lot of regulation, because maybe there are times when we would like to close this hole if it's particularly dry and we're worried about water loss, or open this hole if it's a little bit wetter and we really need to exchange a lot of gases. And so another solution that plants have evolved are stomata. So stomata is plural, stoma is the singular. And what these are is literally they are mouths. Stom in Latin is stome, means mouth. So these are little openings. These are little mouths. And so as you can see in these two pictures, the little openings are created by a pair of guard cells. We have a guard cell here and a guard cell over here. And when the plant is in a very dry environment, our two guard cells can close down and they close that opening. But when there is a lot of water and the plant really needs to exchange gases, the guard cells can expand a little bit and sort of open this hole, as we see in the second picture, and that allows gases to move back and forth. So these are a couple of solutions to our problem of a cuticle, which in itself was a solution to our problem of water loss. So this is how plants can handle desiccation, the drying out of tissues. Finally, let's think about how plants might handle the desiccation of their reproductive structures, particularly the desiccation of their spores. And if you remember and think back to our chara specifically, or more generally even back to our to our charophyceans, they had a material known as sporopollenin. And so in chara, sporopollenin covered the zygote. Now, for plants, what's going to happen is our spores are going to be covered by this sporopollenin coat. And the spores, of course, then surrounded by the sporopollenin coat are going to be very resistant to drying out and very resistant to all sorts of other environmental stresses. And so in fact, what this means is that our spores can exist in the fossil record for hundreds of millions of years, and so we can see evidence of sporopollenin coats going all the way back to about 430 million years ago, and so this again provides good evidence of when plants started to evolve, and particularly when the colonization of land really began to take off in particular areas. All right, so I hope this has provided a good overview of plants and the challenges that they face as they made that the evolutionary transition from water to land. And in our next lecture, we'll talk about our first major group of plants, the bryophytes. So, see you then.